second session. Remember that you can submit questions using the Q&A at the bottom of the Zoom meeting screen, and we'll take all of your questions at the end of the presentation, but feel free to enter them throughout today's presentation. So we are excited to have with us this afternoon Dr. Peter McGuire. Dr. McGuire received his degree with honors from Royal College of Surgeons in Ireland in 2003, following a combined residency in pediatrics and medical genetics at Mount Sinai Medical Center in New York. He remained as an assistant professor in the program for inherited metabolic diseases at Mount Sinai. Dr. McGuire is board certified in pediatrics, clinical genetics, and biochemical genetics. In 2010, Dr. McGuire moved to the National Human Genome Research Institute at the National Institutes of Health to join the Physician Scientist Development Program. He was appointed to the position of tenure track investigator in 2016. Throughout his career, Dr. McGuire has been focused on improving the care of patients with disorders of mitochondrial metabolism. By combining his training in immunology and biochemical genetics, he has fashioned a translational research program to understand the interplay between mitochondrial metabolism and the immune system. As head of the Metabolism, Infection, and Immunity sections at NHGRI, Dr. McGuire and his team study the interplay between metabolism and the immune system in patients with inborn errors of mitochondrial metabolism. Please join me in welcoming Dr. McGuire.
Thank you, Kira, so much for that really wonderful introduction, and greetings to everyone. I hope everyone is doing well and staying safe and staying free of infection. So today I'm going to talk about, and I appreciate the invitation, by the way, to this conference. It gives us a chance to share some of our data and have a dialogue about these issues surrounding infection in patients with inborn errors of metabolism, specifically fatty acid oxidation disorders and other disorders of mitochondrial metabolism. So today I'm going to talk a little bit about infection and why infection is so critical, such a critical stress in patients with inborn errors of metabolism, and how specifically infection can affect fatty acid oxidation, and therefore why patients with disorders of fatty acid metabolism may be at risk during infection for various unwanted outcomes. So we'll get started. So the first thing I just wanted to mention is, as a government employee, I have no conflicts of interest to declare. So in any kind of talk like this, of course, we have to talk about the pandemic in the room, which is the SARS-CoV-2 virus or COVID-19 pandemic. So just to remind people, this was something that, this is a timeline which I'm showing you here, and it's something that we started to hear about back in December of 2019, where the World Health Organization was alerted by China about several cases of pneumonia in a region called Wuhan. And this disease went on later to be named COVID-19. Jumping ahead to about January, this was, within a week's time, essentially, the WHO officials had identified this new virus, SARS-CoV-2, that causes this disease, COVID-19. And it belongs to a family of viruses known as coronaviruses, which includes the previous SARS pandemic, which was back in the early 2000s, as well as MERS, which is a Middle Eastern respiratory syndrome, which occurred in around 2012 or so, as well as it's related to other common coronaviruses, which cause the common cold. So a real range of disease which could be caused by coronaviruses, but this one was a unique coronavirus. Jumping ahead a couple of months to March, the WHO declared COVID-19 a pandemic with more than 100,000 cases and 4,000 deaths in over 114 countries. And on April 2nd, the confirmed cases topped 1 million worldwide. And following that, shortly in April, global deaths due to COVID-19 topped 100,000. So this is a resource, I think, that is really helpful for people who want to follow this pandemic and look at risk on, a international level as well as a local level. So this is the Johns Hopkins University Coronavirus Resource Center. So this is data that accumulates in real time. It is updated several times a day. And these data that we are looking at right now are as of this morning. So there are 15.5 million cases in the world of COVID-19, of which the US has 4 million, which is about 26% of all the cases worldwide. Global deaths are 634,000 plus worldwide, with the US having about 144,000. And that constitutes about 23%. As of this information is always moving, quite quickly. So, there are discussions going on right now with all of the continued outbreaks that are going on and the rise of cases that are happening throughout the United States. There are talks now amongst epidemiologists and physicians, as well as public health officials, with regards to potentially a reboot for the United States, where we would actually change our policies at this point in terms of trying to contain this pandemic. So there's more information to come, and that information, of course, will be disseminated through through the proper public health channels. So a lot of information has come out with regards to this pandemic. It's appeared, in various popular publications, as well as the scientific literature. So this is looking at coronavirus research and how it has expanded essentially since the beginning of the pandemic back in December or so. And you can see this is looking at publications now between February and March of 2020. And the red line indicates the number of journal articles. So these are articles that were peer reviewed and had gone through a rigorous process of peer review before being published, as well as a number of what are called preprints. So this is information that is released prior to peer review, but is done so because of the ongoing pandemic, and that this information may be, is of particular interest to be released kind of early, but it should be noted that this information is not yet peer reviewed. So with this explosion of information, of course, we all have to try and figure out how to sort through it. And that includes families, that includes caregivers, that includes individuals who are affected by, for instance, fatty acid oxidation disorders, as well as scientists themselves. So since there's this kind of deluge of information, whether it be published articles or preprints, it's a challenge for all of us, I think, to try and sort through this information, analyze studies critically, and to be able to disseminate information which is helpful to people. So just as a quick review, let's just talk about what SARS-CoV-2 is. So this is a coronavirus, as I mentioned. And I want to point out that early in the pandemic, there was a little bit of confusion in terms of, this particular virus. First of all, this is a unique coronavirus. This is a family of viruses of which SARS-CoV-2 belongs to. Some of those other coronaviruses can cause things like the common cold and some more severe illnesses. But this particular coronavirus is unique. It causes severe acute, SARS stands for Severe Acute Respiratory Syndrome Coronavirus Type 2. It is a new virus, and as I mentioned, there are other viruses that belong to this family, which range from mild to actually quite severe. So the genetic evidence suggests that this started in animals. One of the animals that is a particularly big source of various types of viruses are actually bats. And bats seem to be able to live in harmony with these viruses. And these viruses, of course, are able to then cross species. And that's how we don't know the exact pathway of how it happened, and may have something to do with open markets in China. But at this point, it has passed obviously into humans, and we have our pandemic. Now, just to point out, this is, not such a matter of semantics, but COVID-19 itself, when you refer to that, that actually refers to the disease. So that's coronavirus disease 2019. So one of the things about this particular coronavirus, and it's unique, I should say we are learning a lot more about this as time goes on, since this is a very new disease and how it behaves. It is very different from the flu in terms of how it enters the body and the different organ systems it can affect, and its severity. So, and that's based on the fact that viruses actually require a key to enter cells. So this here is a cell, this blue part here, and this is the plasma membrane. And on the surface is this receptor called ACE2. Now, ACE2 stands for angiotensin converting enzyme 2. And the point of this is that this is a receptor that is actually targeted by blood pressure medications, okay? So that's why you may hear some stories about individuals with high blood pressure and things like that may be particularly vulnerable to coronavirus. And this disease, those data are not solid yet and still being sorted out, but that's kind of where this came from. And what happens is the virus then takes advantage of this, this receptor to basically make its way into the cell. So this is essentially a keyhole, which the virus can then exploit to get inside the cell. And then once the virus is inside the cell, it then can exploit the mechanisms inside each cell to make more of itself, to replicate itself. And as it turns out, this ACE2 receptor is expressed on many, many different cell types, including parts in the nasal passages in the lungs, which can give you the respiratory disease, in the small intestine, in the heart, in the eye, in the liver, as well as the bladder, the kidney, the pancreas, right, which helps us make insulin, the brain, as well as the prostate and testes, as well as the placenta. So it really has an expression, you know, in many different parts of the body, this kind of, you know, keyhole that the virus can exploit. And it may actually, and the hypothesis at this point is that that's why COVID-19 is such a very, it's a disease like we haven't seen before with many different complications, and we don't understand it completely because there may be multi-system involvement because of the expression of this receptor on many different cell types. So I'm sure a lot of people have heard about this term R0, and R0 is the ability of spread of a virus, basically. So R0 is the average number of people that one person with a virus infects. So for the H1N1 influenza pandemic, back in about 2009, the R0 was 1.2 to 1.6. So in other words, somewhere between 1 to 1.5 people. You obviously can't have half a person. We're talking about very large numbers here, but infects about 1 to 1.5 people, right? Where when we get to things like Ebola, which is a highly contagious disease, but it's also a highly fatal disease. This affects anywhere between 1.6 to 2 people. SARS back in early 2000s, about 2003, had an R0 of 2 to 4, so anywhere between 2 to 4 people get infected from a single individual. Whereas MERS, which is another coronavirus, has an R0 between 2.5 and 7.2. The differences here between this and COVID-19, although they are coronaviruses and they have R0s that are, you know, somewhat in the similar range, is that SARS and MERS were highly fatal. So in other words, the reason that you didn't have such a worldwide pandemic is because there were a lot of people that actually died as a result of this disease. So therefore the spread actually was hindered by the fact that it was highly lethal. Unfortunately, COVID-19 lives in this sweet spot of viruses, right? Where you can have some individuals who are asymptomatic or oligosymptomatic, don't have many symptoms at all. And it has a good R0 in terms of its ability to spread between 2 to 2.5 people. So it really kind of occupies this, as I said, sweet spot for a virus where it is not highly lethal, but is still lethal and is able to spread, and therefore has led to the situation that we are in at this moment. So some of the symptoms that you can have with COVID-19 include things like fever and cough and fatigue and shortness of breath. The loss of taste or smell is something that has been reported in this by individuals who have had this infection, as well as headache and muscle aches, and also GI involvement, which is something that is unique from some of the other respiratory viruses that we normally encounter. So the incubation period is between 2 days to 2 weeks. This is kind of where some of the quarantine guidelines come from. So if you looked in the news recently, New York just issued a list of, I think, about 21 states, which they are advising quarantine if you are traveling to New York State. And that's quarantine of 2 weeks. And that's kind of where this guideline kind of comes from. It's based on the incubation period. And as I mentioned before, the symptomatology of the disease can range from being asymptomatic to mild illness to severe illness. As we're going on, you know, in terms of this disease, we're learning more and more. And some of the manifestations of severe COVID-19 include things like acute respiratory distress syndrome, or ARDS. This is a condition where the lungs essentially fill up with liquid, and it makes it very difficult to breathe. And that's where you hear about ventilators and the need for ventilators. It can cause cardiac injury, it can cause renal injury, and as I showed you before, that ACE2 receptor seems to be expressed on other cell types. Still remains to be determined, how that works in terms of, affecting things like the heart and the kidneys and the brain. But there are some clues out there that people are actively pursuing. One thing that's of particular concern to us, and I think in terms of the type of work that we do with the IEM community, is this idea of hyperinflammation and this multisystemic inflammatory syndrome of children. So I'm going to talk about inflammation in a minute, but basically there is a version of COVID-19 that seems to affect children, and more cases are coming out where it is a multisystemic inflammatory disorder. And you can see here on the right, it represents a disease that looks like something called Kawasaki disease. And Kawasaki disease is a disease that affects, many different organ systems and could lead to things like, conjunctivitis or injected red eyes, cracked red lips, enlarged lymph nodes in the neck or in the groin area, skin rashes. They can also have problems with their heart, where they can have the coronary arteries, which serve the heart. They can become dilated and cause problems later on, as well as inflammation in the heart. So these red ones are what look like Kawasaki disease, or what represents Kawasaki disease. And these other ones in black are some of the additional features of this COVID-19 multisystemic inflammation that's going on in children. So one thing that's important about this is that they're finding out that immune suppression in COVID-19, as well as potentially this disease of children, can actually improve severe disease. And that really highlights the fact that inflammation and the mediators of inflammation, known as cytokines, actually are involved in the pathophysiology of this disease. So what is inflammation? So first of all, inflammation is the immune responses. It's a normal response to infection. You need inflammation. Inflammation is helpful for controlling viruses, for killing virally infected cells, for preventing viruses from getting a foothold in the body. So this is a normal response to infection, and it helps clear the infection. And the mediators of this or the culprits of this are essentially white blood cells, which secrete these little, what we call text messages called cytokines. So these text messages that white blood cells release are necessary for immunity. And it helps the white blood cells talk to each other. It helps them coordinate a response. It helps talk to organs and organ systems about how they should behave during an infection. The problem, though, is when you get something called cytokine storm. So, and that's the hyperinflammation I'm talking about. So in other words, there is a robust release of these cytokines. In other words, in excess of what is actually needed for the situation. And unfortunately, that turns out that it is actually toxic to organs. And those organs can be the heart or the kidneys or the lungs or the liver or the brain. So therefore, in COVID-19, a lot of the problems that are occurring may actually be related to the actual immune response to this virus. So that's why people are thinking of targeting the immune response during COVID-19 illness. And these are severe cases, by the way. I'm not talking about, garden variety, people who have, few symptoms or no symptoms. Okay, so how does this all tie into inborn errors of metabolism and fatty acid oxidation disorders? Well, there is a lot of morbidity and mortality associated with infection in inborn errors of metabolism in general. And there's a term that clinicians use and even families use now because I think they've become so accustomed to it. And this term is called metabolic decompensation. So this can be a life-threatening situation where you can have organ dysfunction or failure, such as liver failure. You can get problems like encephalopathy, where, in other words, problems in the brain. You can get metabolic strokes in certain inborn errors of metabolism where you have death of neurons in certain areas during infection. And then they can have complications or what are called sequelae afterwards. And these complications can persist even after they, so if they survive this episode of infection, because this infection can be lethal, if they do survive, they're often left with, complications afterwards, which either causes their disease to progress or really causes a decrease in their lifestyle, basically. The patients require extensive ICU care, as you can see here on the right, with various things like respirators, and this over here tucked away in the corner is actually dialysis. The patient's being monitored. And simple viral infections can put them in this state. So this even precedes, obviously, COVID-19, where viruses for individuals who don't have inborn errors of metabolism and make them feel, kind of run down, can be a life-threatening situation for these patients. And some of the questions that we've been asking is there a role for inflammation in this? In other words, is inflammation causing, a lot of the complications we may see in inborn errors of metabolism? So some of the changes that you see in metabolism during metabolic decompensation can be traced to the liver. So if you're talking about things like organic acidemias, another type of inborn error of metabolism, you can have acidosis or ketosis or high levels of lactate. Acidosis and high levels of lactate are also shared by mitochondrial disease, may also be shared by fatty acid oxidation disorders. But also in fatty acid oxidation disorders, you can have hypoglycemia. That's because you don't have, with a defect in fatty acid oxidation, you don't have that kind of spare reserve to protect blood glucose. So therefore, blood glucose gets used up, and therefore patients can become hypoglycemic. So fatty acid oxidation helps preserve blood glucose, and therefore, if it's dysfunctional, patients become hypoglycemic. Well, it turns out during respiratory infections, systemic viral infections can actually cause collateral damage to the liver. And since a lot of, inborn errors of metabolism, some of the metabolic problems can be traced to the liver is really a critical organ. So respiratory infections like EBV or CMV, cytomegalovirus, which is a lot like EBV in terms of its infection, herpes simplex virus, SARS-associated coronavirus. This actually, it more refers to the 2002 SARS outbreak that happened, although with emerging evidence now, you could say it also refers to the current SARS-CoV-2, as well as varicella virus and influenza virus. So some of these are, like influenza virus, for instance, are viruses that don't have receptors on the liver. So why is the liver being affected? Why does the liver get damaged during influenza virus infection? And that's one of the things we'll talk about. So in the lab, we use animal models to try and recreate this scenario of viral infection, and we use influenza virus. And even though the COVID-19 pandemic is here, so is influenza. And influenza has been a pandemic for quite some time. It is worldwide. It occurs every year seasonally. It never quite goes away. We do have, unlike COVID-19, we do have vaccinations for it, and create vaccinations every year based on the circulating viral strains. So influenza still remains a highly relevant topic here. So we use mice to study the effects of influenza. So what you can see is we put the mice here in an infection chamber, and we give them aerosolized virus. So essentially they get a lung infection with a mouse-adapted influenza or flu. And over here on the right, you can see these are liver enzymes, right? So liver enzymes are things you can measure in the blood that go up when the liver is unhappy. So you can see here that when animals are fed with, and these are normal mice, these are mice that don't even have an inborn error of metabolism. When you give them influenza infection, their liver enzymes go up, indicating that their liver is either damaged or unhappy or dysfunctional. So what's interesting is that when you take the livers out of these mice, this is a respiratory infection, right? We gave them an infection in their lungs. But when we go and we do profiling of the different messages that are going on inside the liver in terms of how the liver's reacting to this infection, you see a lot of things that are related to infection itself. In other words, recognizing virus and reacting to virus. And that's really interesting because we keep having in our heads that, well, this is a lung infection. Like, why is the liver having something called pattern recognition receptor signaling? In other words, recognizing a foreign invading virus in the body. And why is it activating things like interferons in the liver when this infection is occurring in the lung? So the liver is detecting essentially that there is a viral infection in the body. So it turns out that the liver, besides being a metabolic organ, is actually an immune organ. So there's a lot of cells that have play a role in the immune system in fighting off infections that actually live inside the liver. And those include lymphocytes, or known as white blood cells, There's also something called Kupffer cells. So Kupffer cells are essentially macrophages. They're kind of like the garbage disposals of tissues. And macrophages are in all different types of tissues. And when they live in the liver, they are called Kupffer cells. So they basically eat up the trash inside the liver and help get rid of it, and therefore help control infection and get rid of infection. And I'm bringing them up because when you look in the liver itself, so these are, I just show this because it's very colorful and they're very nice to look at. What we are looking at here essentially is we're counting the number of Kupffer cells in the liver, or these macrophages in the liver, when you don't have an infection and when you do have an infection. And what I want you to focus on is the upper right corner here, where I have the red circle, where when you don't have an infection, the number of Kupffer cells are about 3 or 4%. When you do have an infection, that number increases to about 6%. Now, people are like, oh, well, that doesn't seem like such a big deal. When you're counting millions and millions of cells, this is a big deal. This is almost a doubling essentially of Kupffer cells. And when you take out these Kupffer cells and see, okay, so you have more of them, but what are they actually doing? Well, it turns out that during an infection, they are actually making more cytokines when they are infected, right? So what this means is that they are making a cytokine called TNF-alpha, right? One of these little peptides that is released by Kupffer cells. That is a cytokine that is actually very, very toxic to cells when it is out of control. So TNF-alpha has to be kept in very, very constricted amounts. In other words, you can't release tons of it because it's so cytotoxic. It's toxic to the liver. So once again, stepping back, we have an infection in the lung. We have signals going on in the liver that the liver is detecting viral infection. Well, the question then is the virus actually leaving the lungs and making its way to the liver? So, and the answer is yes. So if you look up here in the left-hand corner, this is it's something called PCR. And what I'm showing you basically in these nice little, pictures here is that when you give an animal this infection, this PR8 influenza infection, you can pick up virus in the blood, and you can pick up virus in the liver. So in other words, the virus seems to be escaping the lung and circulating through the body and making its way to the liver, as well as probably other organs. But what happens in the liver? We have this expansion of Kupffer cells, right? And these Kupffer cells are making this dangerous TNF-alpha. Well, why is that? It's because the Kupffer cells are actually interacting with the virus. They're actually eating the virus. So here on the right in this panel, you can see these are Kupffer cells in culture in a dish. And if we feed them virus that's labeled with a green tag, you can see here that they take up the virus. This is a little Kupffer cell here, as you can see on the right, and inside is all this green. They are actually eating and taking up this virus. And what happens when they eat and take up this virus? They make TNF-alpha, okay? And that's what we're showing here. Kupffer cells, when you give them this virus, make more TNF-alpha. So what are the consequences of this? Okay, so you have an infection that is a lung infection, but actually the virus is able to spread to other parts of the body and activate immune cells, in other words, Kupffer cells, living inside organs, for instance, like the liver. So what's the consequence of this? What are the consequences of these cytokines? What do they do to the liver? Well, they can affect a lot of different metabolic pathways. So you can see here on the left, this is a picture of various aspects of metabolism. You have glycolysis here, which is important for breaking down sugar. You have fatty acid oxidation drawn in a very simple way, which is the breakdown of fatty acids here. And ultimately, this is all passed into the mitochondria, where by the mitochondrial respiratory chain, we make ATP. And ATP is what is the energy currency of the cell. That's what allows cells to work and various tissues and organs to do work. So contained in the mitochondria, as well as the cytoplasm, are all these different pathways. So I'm going to talk about how having an infection can affect some of these pathways, specifically things like beta oxidation, which helps preserve glucose. Okay, first, glycolysis. What happens? So this is important because it does tie into fatty acid oxidation. So when you take an animal and you give it, this is an experiment where you take an animal, and this is a mouse, and you give it something that simulates a viral infection. In other words, you give them something called poly-IC, which is basically a small chemical that when you inject it into animals, the immune system is convinced it has a viral infection. So we did this and we looked to see what happens with glycolysis in the liver when you do this. So in other words, we simulated a viral infection. Now what happens in the liver? Well, you can see here, this is imaging in a live animal, looking at lactate and pyruvate and alanine and pyruvate. So these are, you can take these as kind of indicators of how much glycolysis is going on. And when something is blue, it means cold, it means not much is going on. And when something is red, it means hot, meaning that there is more glycolysis going on. So when we inject the animals with this poly-IC, this viral mimic, to kind of trick the body, the mouse body, into thinking it has a viral infection, you can see here that the liver lights up in terms of this lactate pyruvate ratio and alanine pyruvate ratio. And that's just shown on bar graphs here on the right. So what that tells us essentially is that during an infection, or at least during immune activation, because we're not really giving them an infection, we're just activating the immune system, that there is increased use of glucose in the liver. So that can be problematic, right? Especially in patients with fatty acid oxidation defects, because they don't have that fatty acid oxidation reserve to protect them from using too much glucose. Now, what's interesting here is that if you remember those Kupffer cells, right, we're coming back to them, those cells, those macrophages that live inside the liver that react to influenza infection and make cytokines, when you get rid of them, actually, and you give this drug called clodronate to get rid of Kupffer cells, this signature of using glycolysis a lot goes away. So that teaches us that basically those Kupffer cells help instruct the hepatocytes or the liver cells to use glucose. And they do that by cytokines. They use cytokines like TNF-alpha, for instance. They tell liver cells, upregulate glycolysis, use more glucose. And you can see here that when you get rid of them, that signature goes away. So glucose goes away. And this is just showing here on the right that some of the messenger RNA or some of the proteins that are involved in glycolysis actually go down after you get rid of Kupffer cells. So in other words, you're inhibiting the pathway of glycolysis. What happens to the mitochondria, right? So the mitochondria was at the bottom of my picture I showed before. It's where all of your food, when it's broken down, it feeds into the mitochondria to make energy. Well, it turns out that during this, the mitochondria are actually inhibited as well. So you can see here that complex 1 in the mouse liver, that when you give them this poly-IC to simulate an infection, the ability of complex 1, complex 3 activity goes down. And that can be traced to some of the subunits of complex 1. Complex 1's like a big piece of chewing gum that's like all put together with all these little different proteins. And there are 46 different types of proteins that go into that. And this is just one of them. And that actually goes down. So therefore, complex 1 can't work very well because it doesn't have all of the Legos is probably a better analogy, because they don't have all the Legos to be able to put complex 1 together for it to work efficiently. And it turns out that one of the things that mediates this, as we mentioned, we keep coming back to this cytokine TNF-alpha. So TNF-alpha is made by Kupffer cells. And when you take out liver cells from a mouse and you feed those liver cells with TNF-alpha, you actually see that this is a measure of glycolysis here on the right, these bar graphs. And this is ECAR, which stands for extracellular acidification rate. The only thing you need to know is it measures glycolysis. So when you feed more and more TNF-alpha, glycolysis goes up. But what happens to the mitochondria or oxidative phosphorylation? Well, when you give more TNF-alpha, oxidative phosphorylation goes down. So why is this important? Because when you break down fatty acids, the ultimate acceptor of the basically what's called NADH after breaking down fatty acids is the mitochondria. So if the mitochondria aren't working, breaking down fatty acids is not going to work very well. You're not really going to derive much energy out of that, basically, because the mitochondria don't work very well. Okay, So once again, just reminding you, we talked about glycolysis, which occurs in the cytoplasm, as well as some issues of the respiratory chain inside the mitochondria. And just remember that the mitochondria, right, is the ultimate acceptor of these various products, such basically NADH, from the breakdown of either glucose or fat or things like that. So, okay. So now we're going to talk about specifically beta oxidation. And remember, as many of you may know who are either affected by fatty acid oxidation disorders or care for someone who has a fatty acid oxidation disorder, it helps preserve glucose. So once again, if we go into our animals, these are mice, and we give them an influenza infection. This is just showing, us infecting the animals, and we're matching them for food. So we're making sure each animal is eating the same amounts of food, but one is infected and one is not. And you can see that over 5 days, when we conduct our experiment, both animals lose about the same amount of weight. So basically, this kind of tells us that in terms of food intake, we're matching quite well with our animals. So we can look at the effects of infection specifically. Okay, so when we do that, when we look on the mRNA level, so in other words, the messages that encode various aspects of fatty acid oxidation, we see that they go down during influenza infection. And those are our black bars here. So if it's CPT2 or carnitine acylcarnitine translocase or VLCAD or ACAD or HADHA or ETFA, various aspects of various messages involved in making components of fatty acid oxidation to make sure that it works very well, they go down when you have an infection. And as it turns out, basically the Kupffer cells are also, I showed you before that they're involved in modulating glycolysis. Well, they also modulate fatty acid oxidation. So here, this is doing an experiment, which we do in the dish, where we take Kupffer cells from a mouse liver, and we take liver cells from a mouse liver, and we put them inside. This is a well inside a dish, and they're actually physically separated. So they are not touching each other, but they sit in the same dish. And when you feed them with this influenza virus, what happens is you get the accumulation of fat. So in other words, the breakdown of fat is likely inhibited during this, because we saw that some of the messages involved in fatty acid oxidation go down during this infection. Well, now we're seeing that you have fat accumulation. So it looks like fatty acid oxidation may actually be inhibited in this particular case. And Kupffer cells are mediating that. So once again, if you eliminate Kupffer cells, if you eliminate these macrophages that live in the liver and instruct liver cells how to behave, you recover a lot of the components of fatty acid oxidation. So once again, these are the messages or the mRNA that eventually make up the components of fatty acid oxidation. And you can see the black bars that when you eliminate Kupffer cells with clodronate, you get increases or recovery of CPT2 and ACAD, VLCAD and LCAD and HADHA and ETFA. So in other words, the Kupffer cells are actually like glycolysis. It makes glycolysis go up and it makes fatty acid oxidation go down. Okay, so what's actually mediating this? Well, it turns out that it's actually TNF alpha. So once again, like in glycolysis, TNF alpha was telling liver cells, let's turn up glycolysis, let's burn more sugar. Well, in this particular circumstance, it's telling liver cells, turn down fatty acid oxidation. So you can see here, these are liver cells in a dish, and these are from healthy mice. And when you feed them with TNF alpha, you can see that these speckles here, these red speckles, are a stain for fat, like we saw before. They accumulate fat. And we showed this actually looking at palmitate oxidation in liver cells. So if you take out liver cells from the mouse and you feed them palmitate to break down by fatty acid oxidation, you can see that in a condition where they actually have TNF alpha, or these liver cells are being exposed to TNF alpha, they have a reduced ability to actually oxidize that palmitate. So in other words, break down that fatty acid. Okay, so now back to our immunometabolism. Since as I said, all pathways lead to this funnel, it's a funnel, right? Metabolism is a funnel that ultimately leads to the mitochondrial respiratory chain, which is responsible for ATP generation. And a lot of patients, and Jerry Vockley, I'm sure, has spoken to I've heard him speak about it before. I'm not sure if he spoke about it today, But a lot of patients with fatty acid oxidation disorders also have mitochondrial dysfunction as a part of their disease as well. So we'll talk about the respiratory chain. So in this particular scenario, we took, this is actually a mouse model of mitochondrial disease, okay? So this is a mouse where we knocked out complex 4 of the respiratory chain, so therefore oxidative phosphorylation in these animals does not work very well at all. And what we found is that, so we're gonna start here on the left, and these are electron micrographs of a liver cell. And what you can see is that this is our uninfected scenario, and this is our wild type mouse. This is our mouse that has mitochondrial liver disease. And therefore, when you give it influenza infection, which is PR8 here, the wild type liver, the mouse that doesn't have mitochondrial disease, it looks okay. It looks, you know, kind of like the uninfected mouse. But the mouse that has mitochondrial liver disease, see all these droplets here that are accumulating? That's fat. So dysfunctional mitochondria also has dysfunctional fatty acid oxidation. And by the way, these large balloons here, these darker large balloons, those are mitochondria. So the mitochondria blow up. You can see with the black arrow here, this is pointing to one of them, and they form these mega mitochondria. They're gigantic. And these are fat droplets. And this on the right is just showing the same thing. It's essentially the same picture, but what we're doing here is we're staining for fat. And you're seeing more liver cells here, by the way. This is one liver cell on the left. These are multiple liver cells here on the right. And you can see here that when we, and we think what's mediating all of this accumulation of fat, as we saw, is this cytokine TNF alpha. That's the culprit. That's what Kupffer cells are releasing to cause this damage. So you can see here that when we expose these cells to TNF alpha, the mouse that has mitochondrial liver disease here in the lower right corner, it develops, you know, this accumulation of fat at a much greater rate, much worse than the wild type animals. So these mice that have mitochondrial liver disease are much more sensitive to these effects of cytokines. And we think that sensitivity does carry over into other inborn errors of metabolism, including fatty acid oxidation disorders. Now, once again, when you give this drug to block TNF alpha here, you can see that the fat accumulation goes away. So once again, either targeting Kupffer cells or targeting what Kupffer cells release, which is TNF alpha, you can get rid of this accumulation of fat. And that's just showing the same thing here, where in the lower right panel, we are giving this blocker against TNF alpha, and all this pink stuff, which you can see here on the lower left, goes away when we give this TNF alpha blocker. So targeting the immune system, targeting this particular cytokine can be very effective in ameliorating disease. So basically overall, you know, the reason that any infection, whether it's COVID-19 or influenza or whatever, can be concerning in patients with inborn errors of metabolism and fatty acid oxidation disorder specifically, is that when you do get an infection, it can become bloodborne. It can spread to organs like the liver, where Kupffer cells inside the liver take up the virus, are exposed to the virus, and release these cytokines, which instruct liver cells how to behave. Well, how do they behave? They turn down things like nitrogen metabolism, they turn down fatty acid oxidation, and they turn down oxidative phosphorylation, yet they turn up glucose burning. So in other words, things that you'll see will be like hypoglycemia, and that's a particular concern in patients with fatty acid oxidation disorders. Okay, so what's being done during this pandemic to help combat COVID-19? So first of all, from the NIH, I can talk from the NIH standpoint, there is this active program that's going on, which stands for Accelerating COVID-19 Therapeutic Interventions and Vaccines. And this is a partnership between the NIH as well as industry. And what they did is they divided this into several working groups, including preclinical stuff, which looks at like animal models, and an area of therapeutics to help patients who have COVID-19 or even severe disease and try to get to clinical trials. And then this is assessing clinical trial capacity, and then eventually the hope for vaccines, which the talk at this point is, in the best case scenario, we're talking after the new year. And the purpose of this group is to address what would be the greatest public health crisis of this generation. And it's imperative that all sectors of society work together in unprecedented ways with unprecedented speed. So this is a good website to look at if you want to keep track of these things. This is sponsored by Johnson & Johnson. It's COVID-19 Drugs and Vaccines Tracker. It talks about a number of treatments that are and where they stand in terms of clinical trials. And there are currently two phase 3 trials, such as remdesivir or anti-cytokine treatment. So they're already trying to target the immune system during COVID-19 infection to try and improve patients' outcomes, as well as a number of vaccines. And this is taken from a little bit, probably about 2 or 3 weeks ago. So this may be even updated beyond this at this point. So what can patients with fatty acid oxidation disorders do to mitigate their risk of infection? So these are some of the CDC guidelines, and I put the link at the bottom for those of you who wish to look them up. So there currently is no vaccine, as everyone knows, and it's being worked on. And there are some promising reports out of Oxford, as well as some other companies who are developing vaccines. But the best way to prevent illness is to avoid being exposed, right? It's understanding that there is person-to-person spread, and it happens with close contact, and that's why the 6 feet rule comes in. Or 1 meter, depending on your location. Respiratory droplets are produced, when an infected person coughs or sneezes or talks, and these can land in the noses or mouths of people and lead to infection. And most importantly is that these are about the asymptomatic individuals, that some individuals who are asymptomatic may actually also be spreaders. Washing your hands, you've probably heard this before, soap and water is actually quite effective in terms of, trying to keep your hands clean. And this should be after you're in a public place or blowing your nose, coughing or sneezing. And if soap and water is not really available, hand sanitizer has to have at least 60% alcohol in it to be able to be effective. And of course, we've all heard about avoid touching your eyes, nose, and mouth with your unwashed hands. Avoiding close contact is important. Especially limiting contact with others as much as possible, and close contact, especially with people who are sick, and putting distance between yourself and other people. So face masks. So basically face masks serve 2 purposes. The real main purpose is to help prevent the spread of to others, basically. And this is even for people who don't feel sick at the time. And at this point, the CDC is recommending that everyone wear a cloth face covering in public settings. And as I said, the cloth face mask is meant to protect other people, but also, it does offer some protection to yourself. And this is, it says here about not using a face mask meant for a healthcare worker. That, of course, is the purpose of that, of course, is for the personal protective equipment that is needed for healthcare workers who really are on the front lines of this. And then, of course, keeping about 6 feet away from yourself and others. Well, between yourself and others, excuse me. Okay, covering coughs and sneezes. This is something that I know, in the metabolism clinic is addressed a lot. And for younger kids, we encourage them, to cough into their elbow. So always cover your mouth and nose, throw your tissues in the trash, and then wash your hands after coughs and sneezes. Cleaning and disinfecting is an important part of this as well. Touched surfaces. Things that, like tables and doorknobs and light switches, things where virus could potentially be passed. If surfaces are dirty, clean them using detergents or soap or water prior to disinfection. And then use a household disinfectant. And there's a list here that you can find of EPA registered household disinfectants. So how effective are these practices? And I think that's something that is important to reiterate. So this is a meta-analysis that came out in the Lancet recently that looks at some of these practices of social distancing and masking, et cetera, and actually found that these practices were actually quite effective in terms of preventing the spread of infection. But if that didn't convince you, healthcare workers probably, this is a really good study actually that came out in JAMA recently, looking at healthcare workers, right? So these are individuals who are exposed to, COVID-19, on a daily basis. So this looks at the implementation of universal masking procedures for healthcare workers. And you can see here that and this is on the axis here is the number of COVID-19 or SARS-CoV positivity in a hospital. And the dots, the bigger the dot represents how many tests were done per day. So in the beginning, they're doing a lot of tests. You can see that this is rising. They institute the universal masking procedures for healthcare workers. And you can see then afterwards, you actually have start to have a drop and a continuing drop essentially in the level of SARS-CoV positivity. So in the worst case scenario where the highest risk of exposure takes place, masks actually are quite effective. So in summary, viral infections and inborn errors of metabolism in general, inflammation during viral infections can have a negative impact on hepatic or liver metabolism, and that can be a contributing factor to metabolic decompensation. Remember, the liver is important for maintaining blood glucose, especially in disorders of fatty acid oxidation. So it's important to, our goal is to try and keep patients healthy and keep them from getting infections. And then with regard specifically to SARS-CoV-2, the mitigation procedures that are currently being recommended for COVID-19 actually are quite effective. The data are supporting, these procedures as hand washing and social distancing, and especially wearing a mask. So I'd like to say thank you to everyone. This is just some of the people who are in my group and are responsible for some of the studies that you see, and I'd be happy to take any questions.
Wonderful. Thank you so much, Dr. McGuire. This is all really, really amazing information. And as we know, it's in the top of mind for everybody. And there's so much information out there. It's good to hear directly from you and kind of put everything in perspective. So a few questions.
Yes.
Given metabolic decompensation, have you noticed a tendency for patients with FAODs to not be infected as often to compensate?
Yeah, so that's a difficult question to ask because, to be honest, we really don't follow at the NIH Clinical Center. We haven't been following FAOD patients. And this all kind of stems out of this idea of a paper that was published a couple of years ago, which suggested that fatty acid oxidation is important for immune cells to function. And basically, I'm not saying those data are not true, but there's still some debate about whether patients with fatty acid oxidation disorders or any mouse models or et cetera get infections more often or less often, or their ability to fight off infection is hampered. When a couple of years ago, when I was seeing some patients with fatty acid oxidation disorders, we did have people come and see us who had problems with immune function, but I don't have a big enough collection of individuals to say that this is something that's widespread.
Right, great. Thank you for that. Do you think that FAOD kids are at a greater risk for the multi-system inflammatory system due to their already increased inflammation?
Yeah, so that's interesting. So I know Dr. Vockley has some data which I've seen him talk about, which suggests that patients may have enhanced inflammation. And yeah, so I can't say specifically that they are more prone to this systemic inflammatory. No one has data for that yet, so that's kind of hard to answer. But what I can say is that we do know that even at what's considered normal inflammation, in disorders of fatty acid oxidation, that normal level of inflammation, which is supposed to be there for fighting off viruses and stuff like that, that can even be deleterious because the tissues that are affected by inborn errors of metabolism, whether it's fatty acid oxidation disorder or mitochondrial disease or whatever, don't react to those cytokines very well, actually. They become more dysfunctional. The tissues may even die. So while I can't answer directly whether the patients have that, I do think inflammation is potentially a concern in this population, and therefore trying to keep individuals as healthy as possible is the best practice we can do right now. Okay,
great. Is it true that patients with long chain FAODs have revved up immune systems already? I'm worried that the COVID induction inflammation could make it worse. Oh, it is true. I'm sorry. This is Dr. Vockley.
Yeah.
He's jumping in and providing a response. So he's saying it is true. The patient with long chain FAODs have revved up immune systems already. I'm worried that the COVID induced inflammation could make it worse. So he's adding to your answer.
Yes, exactly. And as time goes on, I mean, it's one of those scenarios where you hope you don't get those data, right? Because that then implicates that people are getting sick and getting really, really sick with COVID-19. However, you know, these are things that are being looked at by people like Jerry, et cetera. So these things will be very helpful, you know. So if you do become ill, I do encourage everyone on this call to contact their providers, because the more information we can get about this, even if you weather it well, still let your provider know, because I think what we can start to do as a community is learn who is more susceptible and who is not.
Right, right. Great, thank you. Yeah, it's important to collect that information.
Yes.
2 more questions, and then we will sign off and get ready for the next session.
Yes.
What are your thoughts about the importance of getting a flu shot and potentially being exposed to COVID-19, and how important is it to get a flu shot as we approach the fall season?
Yeah, I mean, we are big advocates of the flu shot, you know, as well as other recommended childhood vaccinations. The flu, of course, is not going away, unfortunately, right? So as the fall approaches, so flu season, you know, it varies a little bit, but it can start as early as, you know, October, September, and go as late as April. Flu never goes away, right? It's just that the reason it increases during that kind of wintertime period is because a lot of us are indoors and we're in close quarters, and spread happens a lot easier. So we do encourage people getting the flu shot every year, because, you know, flu itself, it can have very detrimental effects on patients with inborn errors of metabolism.
Yeah, great. Thank you. So last question. We were told by our daughter's geneticist that AST and ALT also rise when muscle is breaking down in our LCHAD child. How do we determine if AST and ALT are on the rise because of liver damage or on the rise due to muscle breakdown?
Yeah, so there are other, I mean, there are obviously other measures you can look at to look at muscle more specifically, like CK, for instance. And the liver, too, has a lot of other functions, right? So liver is one of the main organs for getting rid of lactate. Liver is one of the main organs for getting rid of ammonia, you know, and maintaining blood glucose. So there are a lot of other, you know, in other words, whenever we assess an organ or an organ system, we're never looking at one specific measure. You're kind of looking at more of a complete picture. So yes, that's absolutely true that enzymes that normally you would think of as liver enzymes can also go up with muscle damage. However, there are other things that you look at as well in terms of assessing organ health.
Okay, great. So thank you, Dr. McGuire. I can't thank you enough for sharing this information. As I said, you know, COVID-19 is on the top of everyone's mind. And I think everyone, every household, every family is struggling to navigate and to know what's the right decision and, you know, how do we best protect all of our families. So we really appreciate you taking this time to share this information with us and giving us the tools that we need to make the best decisions for our families. So we truly, truly appreciate that.
Oh, I appreciate you having me and allowing me to share some of that information.
Yes, it's wonderful. So we, just for the attendees on the call, the next session will be with Annette Hines and preparing for a new normal in COVID-19. So we're going to sign off of here and ask everyone to go into the auditorium to the schedule and go into the next session with Attorney Hines, and we will kick that off in just a few moments. Have a great day, everybody, and we'll talk to you in just a minute.